Welcome to the Chemistry, Biology and Math Revision Hub. Today we are doing the Pearson Edexcel Level 3 GCE Biology B Paper 1 for June 2022. This is the Part 2 video. I'll put the link to the Part 1 and Part 3 videos below in the description box. Let's begin with question 6. Question 6. The lipid content of the cell membranes of prokaryotic organisms changes in response to changes in the environmental temperature. Phospholipids form a bilayer in the cell membrane of bacteria. Describe the structure of a phospholipid. A phospholipid is made up of glycerol, which is attached to two fatty acids by an ester bond, and the phosphate group is attached to the glycerol. Next, they say, the cell membranes of most organisms belonging to the domain archaea are lipid monolayers. The lipid that forms this monolayer is a bipolar lipid. Explain why a bipolar lipid is a suitable molecule to form the cell membrane. Now we know that a bipolar lipid will of course have two polar groups. So here I said a bipolar lipid has two polar groups. One polar group will interact with the aqueous environment on each side of the membrane, and the phospholipid part of the bilipid will provide a barrier to prevent movement of polar and ionic compounds in and out of the cell. Moving on. Question C says, the percentage of branched-chain fatty acids in membranes of bacteria that have different optimum growth temperatures was investigated. The graph shows the results of this investigation. So here on the vertical axis, we have optimum growth temperatures of bacteria, and on the horizontal axis, we have the percentage of branched-chain fatty acids. Down here, they say calculate the mean percentage of branched-chain fatty acids in bacteria whose optimum growth temperature is less than 20 degrees. This is where the 20-degree mark is. So we see these are the ones that are below that. So there is one at 0, you can see that, and that at 0. There is one at 2 and one at 4. So adding everything up, which is a 0, a 0, a 2, and a 4, and dividing by the number, which is 4, gives us 6/4, which is 1.5%. Here they say analyze the data to describe two conclusions that can be drawn from this investigation. So here we see there is a general trend. The higher the percentage of branched-chain fatty acids, we see the higher the optimum growth temperature, and as the percentage of branching increases, the range is going to be spread out. As we can see here, there is a wider gap between the lowest and the highest, with a higher percentage of branching, while at lower, there is a lower range between the highest and the lowest. The greater the percentage of branched-chain fatty acids, the higher the optimum temperature, and the bacteria with higher percentage of branched fatty acids have a wider range of optimum growth temperatures. Moving on. Here they say a change in temperature can affect the permeability and fluidity of the membrane. Explain why it is important that the lipid composition of the membrane of prokaryotic organisms changes if the temperature changes. Now we know the purpose of the membrane is to control what enters inside and what leaves the cell. If the temperature changes, there is going to be a change in permeability, and if the membrane does not adjust itself to accommodate the change, then there is a chance that molecules will enter the cell without control. So here I say the membranes control what enters or leaves the cell, which is membrane permeability. And we know the higher the temperature, the higher the permeability of the membranes. And this helps to provide nutrients like glucose and oxygen, as well as the removal of waste products. So the fluidity of the membrane must be maintained in order to achieve this. This brings us to the end of question 6. Let's continue to question 7. Question 7. Macrophages are involved in response to infection. Macrophages engulf bacteria by surrounding the bacteria with pseudopodia. The image shows a macrophage forming pseudopodia around some bacteria. So you can see the bacteria here, and this is the pseudopodia formed by the macrophage around the bacteria. Down here they say, the flowchart shows one theory for the formation of pseudopodia. In step 1, antibody binds to the cell surface of the macrophage. And then step 2, the fibrous protein in the cytoplasm close to the antibody binding site break down to form soluble components. Step 3, water flows into the part of the cell where the soluble components are located. Step 4, pseudopodia start to form around the bacteria. Here they say which diagram shows one antibody binding to the surface of a macrophage. The answer is going to be a D, because if this is the macrophage, the antibody binds using a single link, while it binds using two links onto the antigen. Next part they say actin is a fibrous protein. Which row of the table describes the breakdown of actin? Since actin is a fibrous protein, of course, the monomer formed is going to be amino acids, and the process of breaking down the bond is going to be a hydrolysis reaction because water is added to the bond as the bond breaks. So here the answer is a B. Here they say, explain why water flows into a part of the cell where the soluble components are located in step 3. We know that water is going to flow into the cell by the process of osmosis, and therefore there should be some soluble components within the cell that can dissolve in order to cause osmotic pressure, to cause the movement of water from a region of high water potential into the cell where there is low water potential. So here I said, presence of soluble components decreases the water potential, so water moves towards that part of the cell by osmosis. So from the section with higher water potential to one with a lower water potential. Moving on. Describe the events that take place resulting in T helper cell activation following the formation of pseudopodia by the macrophages. We know that the macrophages, of course, engulf the bacteria, and they will release lytic enzymes into the phagosome. Digestion of the pathogen takes place, and then the antigens are going to be sorted out and attached onto the MHC protein, and these antigens are going to be presented. So antigens from the pathogens are attached to MHC protein, and the macrophage carries out antigen presentation to the T helper cell. Then the T helper cell binds to the antigen on the antigen MHC complex on the macrophage, and this activates the T helper cell. Moving on, Part B says, phagocytosis by macrophages is affected by a number of factors. Microorganisms with a fluorescent green dye attached to them were used to measure phagocytosis by macrophages. The microorganisms were added to the macrophages and incubated, and the macrophages were then washed, and the extent to which they glowed green was determined. The extent of the green glow is proportional to the number of microorganisms engulfed by the macrophages. The graphs show the results of investigations into phagocytosis by macrophages. Extent of green glow, and here we have the length of the investigation. We see as the length of the investigation increases for all concentrations of microorganisms, the extent of green glow increases. Also here we see the extent of green glow as the length of investigation is varied, but here the number of macrophages per unit. Also we see that overall as the number of macrophages increases, when the length of the investigation increases, the extent of green glow is increasing as well. Here we have the extent of green glow on the vertical axis and the source of the macrophage on the horizontal axis. We see some from the lungs, some from the abdomen, and those from the bone marrow. We can see there was more green glow with those from the bone marrow. The last result shows the extent of green glow versus the concentration of ATP synthase inhibitor. We can see as the concentration of ATP synthase inhibitor increases, the extent of green glow is decreasing. So here they say analyze the data to discuss the factors that affect phagocytosis by macrophages. So I said from the first graph, as the time of the investigation increases, more bacteria are taken up by macrophages. Also, the higher the concentration of bacteria, the more they are taken up. However, the results are only observable after 10 minutes. If I take you back here, you can see it's only after 10 minutes that we can begin to observe the results. Although we see the higher the length of the investigation and the higher the concentration, we see the higher the extent of green glow. From graph 2, the more the macrophage is present, the more the uptake of bacteria, and for all concentrations of macrophages, there is no difference in the extent of green glow until 30 minutes. After that, great difference exists only when 100,000 macrophages are present. I will show you that here. We can see this is a greater difference that is going to be observed, but there is almost no difference until we see here. That is where they begin to separate, and we can see a distinctive difference with this, but these remain close until about 50, so there is great observation with higher number of macrophages. From graph 3, the source of the macrophages affects how they are taken up, meaning phagocytosis. We observe that bone marrow macrophages take up more of both types of bacteria, and uptake of bacteria by macrophages depends on the type of bacteria. Bone marrow macrophages take up more bacteria B than bacteria A. From graph 4, the higher the concentration of ATP synthase inhibitor, the lower the uptake of bacteria. And this is because after 100 AU, the inhibitor stops ATP production, so less ATP is available for phagocytosis. So this brings us to the end of question 7, as well as to the end of the second video of this paper. Thanks for being with us. Do not forget to subscribe. See you in the next video. Bye-bye.